Hey there, science fans! Today we're diving into the fascinating world of proteins, the building blocks of life. And to study these tiny but mighty molecules, scientists use a powerful technique called 2D gel electrophoresis. Imagine a giant chessboard where each square represents a different protein. That's essentially what a 2D gel is. It's a technique that separates thousands of proteins from a complex mixture, like a cell extract. This separation allows scientists to study individual proteins in detail. Pretty cool, right? 2D gel electrophoresis is like a protein detective helping us identify, quantify, and analyze proteins. This information is invaluable for understanding various biological processes, disease mechanisms, and drug discovery. Now let's break down how this amazing technique works. 2D gel electrophoresis involves two separation dimensions. The first dimension is called Isoelectric Focusing, or IEF for short. Remember that each protein has a unique charge based on its amino acid composition. IEF exploits this property by separating proteins based on their isoelectric point, pi. The pi is the pH at which a protein has no net charge. It's like its comfort zone. Think of a racetrack with different lanes having varying pH levels. In IEF, proteins are placed on this track and an electric current is applied. The proteins migrate until they reach the lane corresponding to their pi, where they become neutral and stop moving. This first dimension of separation creates a strip of proteins arranged by their isoelectric points, ready for the next step. The second dimension of separation is called SDS-PAGE, which stands for sodium dodecyl sulfate polyacrylamide gel electrophoresis. Sounds complicated, but it's actually pretty straightforward. In this step, the protein strip from IEF is placed on top of a polyacrylamide gel, kind of like a jello-like substance. But before that, the proteins are treated with a special detergent called SDS. SDS is like a molecular straightener for proteins. It unfolds them and gives them a uniform negative charge. This ensures that the proteins will now separate solely based on their size, with smaller proteins zipping through the gel faster than larger ones. Think of it like a race through a crowded obstacle course. Smaller, more agile proteins can navigate the obstacles faster than larger, bulkier ones. At the end of SDS-PAGE, we have a 2D gel with proteins neatly separated into thousands of spots. Each spot represents a different protein defined by its unique pi and molecular weight. Section 4. The 2D gel electrophoresis process, a step-by-step guide. So let's recap the entire process step by step. First, we need to extract proteins from our sample, whether it's cells, tissues, or any biological material. This is like squeezing the juice out of a fruit to get all the good stuff. Next, the protein extract is loaded onto an IEF strip and an electric current is applied. Proteins migrate to their isoelectric points, creating a separation based on charge. Then the IEF strip is placed onto an SDS-PAGE gel and the proteins are separated further based on their molecular weight. After separation, the proteins are visualized using stains or dyes that bind to them. This creates a pattern of spots on the gel, each representing a different protein. Finally, individual protein spots can be cut out from the gel and analyzed using mass spectrometry. This powerful technique helps identify the proteins present in each spot. Section 5. Visualizing and identifying proteins. Visualizing the separated proteins on the gel is like revealing a hidden treasure map. Several techniques can be used, but the most common one involves staining the proteins with dyes that bind to them. These dyes can create a colorful pattern of spots on the gel, with each spot representing a different protein. The intensity of the spot reflects the abundance of that protein in the sample. To identify specific proteins, individual spots can be cut out from the gel and analyzed using mass spectrometry. This technique is like a protein fingerprint scanner, providing information about the protein's mass and amino acid sequence, allowing scientists to identify it. By comparing the protein profiles of different samples, researchers can gain valuable insights into various biological processes. Section 6 and 2, D-gel electrophoresis, a powerful tool for scientific discovery. 2. D-gel electrophoresis is an incredibly powerful tool that has revolutionized the field of proteomics, the study of proteins. By allowing scientists to separate, visualize, and identify thousands of proteins simultaneously, This technique has opened up endless possibilities for scientific discovery. From understanding disease mechanisms to developing new drugs and therapies, 2D gel electrophoresis plays a crucial role in advancing our knowledge of life's intricate processes. It's like having a powerful microscope that allows us to peer into the complex world of proteins and unravel their secrets. So the next time you hear about groundbreaking discoveries in medicine or biology, remember the incredible power of 2D gel electrophoresis, a technique that continues to illuminate the hidden world of proteins and their impact on our lives.